Welcome back to Biomindscape. Today we are going to know about the chemosmotic theory that was proposed by P. Michael that explained that ATP is formed by using a proton, that is a hydrogen gradient, across the thylakoid membrane when the light energy is absorbed by the PS2 and PS1. Here we can see in this diagram there is PS1 and PS2. Electrons are getting excited, they are moving through the electron carrier like the cytochrome B6F complex, and during this process water is splitting, that is photolysis is happening and releasing the oxygen. The electron and proton they are released, and these protons are accumulated inside the thylakoid lumen that create a high concentration, while the stroma it have a lower concentration, and they together form a proton gradient. This gradient act like a storage energy. Proton then move back to the lumen of the stroma through the ATP synthase complex, that is CF0, from the CF0 to CF1, and this movement then release energy that is used to convert the ADP to the ATP. The ADP is getting converted into the ATP, and this process is called chemosmosis. So in terms of the energy requirement, the C3 plants use 3 ATP and 2 NADPH to produce one oxygen molecule, whereas C4 plants they require 5 ATP and 2 NADPH, meaning the C4 plant need more ATP for the same process. Now let's see the working of this diagram. So here we can see the splitting of water molecule. So as PS2, here in the inner side of the thylakoid membrane, the light energy hits the P680, that is PS2, which splits the water H2O into the proton, that is H plus ion, and electron, that is E minus, and oxygen O2, which results in the hydrogen ion. They are released directly into the lumen that increase the proton concentration inside. Then the proton pumping that is occurring here between the PS2 and the cytochrome B6F complex. Here as the electron it is moving through the chain, a carrier called the plastoquinone PQ picks up the hydrogen from the stroma outside to move the electron. When it has a electron to the cytochrome B6F, and it releases those H plus hydrogen into the lumen, which results in active pumping the proton from outside to the inside. Then the third step is removing the proton from the stroma, that is PS1. Here on the outside, the stroma side of the membrane, working is like electrons they reach the P700 PS1 and they are passed to the NADP, and an enzyme NADP reductase takes the hydrogen from the stroma. What is happening here? On the outside there is a stroma side of the membrane, and the electron is reaching the PS1 and NADP, an enzyme NADPH reductase. That reductase takes the hydrogen from the stroma. It is taking hydrogen from the stroma and it is reducing the NADP to NADPH. Here we can see in this diagram the NADP it is getting converted into NADPH. And how this happens? By taking an hydrogen ion. It is taking hydrogen ion out of the stomata, and the difference between the inside, that is a lumen, and outside there is a stomata, and they become even greater. Now how this working? Because of the step 1, step 2 and step 3, the lumen become very acidic. There is high amount of hydrogen ion, because of that it is very acidic, and high hydrogen ion in the stoma become basic, that is inside the stoma the hydrogen ion are not there, they are low hydrogen ion are present in the stoma. That results in creating the electrochemical potential gradient. The electrochemical potential gradient we can see here, that is an yellow arrow. So ATP synthetase, here what's happening? Through the ATP synthetase, the CF0 that is getting and CF1 complex, they are here present. The proton rushes back to the stroma to the CF0 channel, and this flow of energy causes the CF1. There is an energy flow, and that is causing the CF1 and headpiece to change its shape. That result in conformational change and provide an energy to link ADP plus ionic phosphate, and together ADP plus ionic phosphate they will form the ATP. So light is used to build the proton pile inside the lumen. So inside the lumen, the proton are built, and the only way out of this proton is through ATP synthase. That is turbine which spins to create the ATP. Thanks for watching. Do like, share and subscribe Biomindscape.